welcome, and thank you for joining us today for our series, which is the Sight and Sound Bites series, webinar series from the Eye and Ear Foundation. This lunchtime educational webinar series highlights the research and clinical innovations at the University of Pittsburgh School of Medicine, Department of Ophthalmology today. Our topic is going to be retinal degenerations, light at the end of the tunnel. I am Lawton Snyder. I'm the CEO of the Eye and Ear Foundation. The Eye and Ear Foundation supports research to advance care for vision, hearing, balance, voice, and cancers of the head and neck. We work closely with the two world-renowned departments of ophthalmology and otolaryngology at the University of Pittsburgh. The funds we provide annually to those two departments stimulate and advance groundbreaking research. And that support is only possible because of philanthropic donations, which we bring in through the Eye and Ear Foundation to support those departments. Thank you for all of those who have been supporting us over the years, and thank you for the new people who are coming on today to hear about the work that's going on. Before we start today's program, I need to go over a few housekeeping items. This is Zoom program. It's similar to Zoom that you may use to chat with friends and family, but this is a webinar program, and we, for the purposes of today's webinar, disabled chat. If you're used to chatting, that won't be available to you, but we do welcome and encourage questions and answers. So if you want to ask a question at any time, there's a little Q&A bubble at the bottom. You can click onto the Q&A bubble and you can type your question in anytime during the program. We will hold questions until the end, and I'll read the questions. I will ask that we refrain from very personal health questions, and I apologize if I do pass some of those over. It's really just because we want to make sure the questions we're answering are relevant to the entire audience. So what we will do, though, and we'll in every case we try to make sure if anybody has any questions that our clinical team can answer, we will we'll send those to those people if it's appropriate. Tomorrow you'll receive a survey via email to provide us with feedback. Thank you for helping us with that. You'll also be added to our email list to receive future webinars, which again, we're doing this bi-weekly series. I'm excited about today's program because we have two fantastic speakers. Today's speakers are Dr. Jose Elaine Sahel, our distinguished professor and chairman for the Department of Ophthalmology, the University of Pittsburgh School of Medicine, exceptional class professor, Sorbonne University in Paris, and the Eye and Ear Foundation endowed chair. He is going to be joined by Dr. Leah Byrne, an assistant professor in ophthalmology at the University of Pittsburgh School of Medicine, director of the Gene Therapy for Retinal Disease Laboratory. I can tell you Dr. Byrne was the second person Dr. Sahel recruited here from Berkeley. Very, very bright scientist who's been doing some exciting work, and you're going to get to hear a bit about that and again about this topic, which I know is near and dear to Dr. Sahel and his interest. And again, it'll be a fantastic program. Thank you. Please take it away. You're muted, Dr. Sahel.
I did unmute.
Okay,
can you hear me?
You're good.
Yeah.
Yep.
Okay. So this is Jose Sahel. I'm very pleased to share with you the excitement we have on during the fight against a blinding disease that has been lacking any therapy for so many decades and is still lacking therapies in many cases. But we have made a lot of progress over the past decades, and obviously we won't have time to get into all of it, but we need to share with you a lot of the excitement we have with that. I'm very pleased that Leah Byrne, who is an amazing gene therapy expert, is joining me for this webinar today. We have many more people in the department that work on this topic, and hopefully we'll share with you also in the future. So we call that light at the end of a tunnel because, as you may know, in retinitis pigmentosa, vision becomes like a tunnel vision, and the tunnel becomes narrow and narrow over the years, and eventually several patients are losing central vision. And now there is some hope to protect patients from losing their vision, not totally, but in part, and potentially also to restore some vision. This is work in progress, and you need to be very careful. Anyone who is claiming that he's curing blindness is certainly someone you should not trust, because this is not yet there. We are working on it very hard, and it may happen in the near future, but a lot remains to be done. So I'll go on to the next slide and just summarize for the people that are not familiar how light is captured. It gets through the eye and through the lens, and is captured at the level of photoreceptors in the depth of the retina. Photoreceptors are transforming light into signal, and this signal is then processed in the retina and back to the brain. The place where we see is the brain, actually. The retina and the rest of it is really processing the information. And this processing starts in the retina, that is part of the brain, in the outer retina, the inner retina, and then the sequence of events leading to the form vision, which is a miraculous thing we have every microsecond, is happening at the level of the brain in a continuous mode. Unfortunately, there are conditions that are affecting that, and we rely very heavily on photoreceptors because photoreceptors contain the pigments that are necessary to capture the light and to trigger the electrical reaction. So they are the ones that enable us to see the light. As you know, the light is only a small part of a spectrum of radiations, and we don't see the other radiation. We don't see radio, we don't see microwave, we don't see X-rays. We see only the visible part of the light. And because we have these photoreceptors, the rods that enables us to see at night at very low levels of light, and the cones that are seeing during the day and anytime we have enough light for that, and are also accountable for form vision and color vision. The retina is very particular because we benefit from both a very high resolution part of the retina, which is called the macula in the very center, but has a very high density of cone photoreceptors and also a very high density of neurons processing the information. The rest of the retina is enabling us to detect everything that happens in the periphery, like obstacles. So we are sharing the high resolution vision like some birds have, and the peripheral vision that many other species have. We have a very sophisticated system that can work both at night and during the day, and can work both in the periphery and the center. Everything is relying on these photoreceptors, and these photoreceptors are a very complex cell type that has a lot of events occurring, a lot of proteins involved in the transduction, which is the transformation of light into electrical signal. And when photoreceptors are not functional, none of this is happening. So just as a reminder about central vision, it is the very center, what we call the fovea. This is where the cones are concentrated, and this is where we have the highest resolution of vision. If you look at this painting by Renoir of Mrs. Henriot, you can see that the face of this woman is high resolution, whereas the rest of the body is kind of fuzzy. This is what happens when you fixate, let's say, our eyes. This is high resolution, the rest is very fuzzy. And unless you move your eyes in another direction, it will remain so. So we rely only on the central part of a retina for high resolution vision, and the rest of the retina enables us to see at night and to see with lower resolution in the range of 2400 or below. So what happens is that there is a group of conditions that does affect either the center or the periphery. We had a webinar a few weeks ago about age-related macular degeneration. In age-related macular degeneration, there is a loss of the central part of the retina, the photoreceptors mainly, and people are no longer able to see high-resolution details, in that case, the face, for example, so everything is kind of fuzzy. In the group of conditions we are going to discuss today, what we call retinitis pigmentosa or road cone dystrophies, patients are losing dark adapted vision initially, then they lose peripheral vision, and they have what is called a tunnel vision, where they can see with very good resolution the details in the center, but they don't see at night and they don't see in the periphery. And many patients, on average, can be what is called legally blind, which is not full blindness, but not being able to perform in many things at the age of 40. Some it's much later, some it's much earlier, and sometimes it's at birth. It's called Leber's congenital amaurosis. So patients affected with retinitis pigmentosa are affected with a disease that is affecting the periphery of the retina, and there is a lot of pigment in the fundus, and this is why Donders was a Dutch physician, thanks to the discovery of the ophthalmoscope in the mid-19th century by Van Grief, was able to describe the disease as retinitis pigmentosa with a lot of pigment. Actually, the pigment is a secondary phenomenon. The main thing is the loss of photoreceptors and the sequential events that lead to blindness. This is a picture of what a patient with retinitis pigmentosa is able to see. Initially, the patient is losing dark adapted and peripheral vision. This is what we measure in the clinic, that is called the visual field. And patients have only what is called the tunnel vision, as you can see here, and they want to see the beautiful cathedral in Strasbourg, where I was working for many years. They see only a small part of it. So to really be able to see something significant, they would have to move their eyes and to really build an image out of that. Not easy. Worse is that over the years, this tunnel becomes more and more narrow, and eventually patients can lose totally vision. This is what we want to exemplify in the sequence of events. Initially, vision can be pretty normal. Then patients are losing dark adapted vision. Then the next stage is the tunnel vision, and the late stage is loss of central vision. This corresponds to a sequence of events in the retina. Initially, the rod photoreceptors, the numerous cell photoreceptors that work at night, are able to perform at low levels of light, are affected with mutations. And these mutations, which are many proteins, there are hundreds of proteins in photoreceptors, are inducing a loss of function and the loss of the seeing part of photoreceptors and the degeneration. And because of that, the rods are dying progressively. There is 130 million rods in the retina, and most of them are dying progressively. And as a secondary event, the cones, but the cells that are so important for central vision, seeing, reading, recognizing faces, are degenerating, and then patients are losing central vision. So the sequence of events is that you have many mutations leading to the loss of the rods. And as a secondary event, in most cases, some patients have direct mutation in cones, but in most cases they don't. Then the cones are dying, and this is a secondary event. We'll get back to that in the next part of the presentation soon. So this is what's happening to the patient, and it corresponds to what we see on the fundus. So this is the pigment that I described, and initially you have a sparing of a central part of a retina. We are using many ways to document that in patients. We are using many technologies. If you come to the clinic, you know that we are doing a lot of testing for you. We are testing the accumulation of pigment in the retina, the health of photoreceptors using different types of imaging. I'll show you a few of them, including new technologies that we are developing. We are measuring the activity of a retina by electrical measurement, that is called the electroretinogram. And we are assessing the visual field to determine the loss of peripheral vision and potentially also central vision. This is also another type of disease that belongs to the same family, which is not affecting the peripheries, affecting the center. It's called Stargardt disease. I think we'll have a dedicated seminar on that disease. It's a different type of disease, but it's also a genetic disorder that affects the center of a retina and eventually also leads to very severe loss of vision. But for today, we'll focus on retinitis pigmentosa. We are able to image, as I said, photoreceptors in the retina, and we benefit from technologies that were discovered over the past few, I would say the past decade and before. One technology that was discovered by a group of scientists, including the previous chairman of the department, George Schumann, is optical coherence tomography. It's like a CT scan, a scan that is using X-ray, but in that case, it's using the light to perform an optical section of a retina. It's totally non-invasive. It's using infrared light, and you get a picture of the thickness of a retina. You see the layer of photoreceptors and all the layers of a retina that contribute to vision. And there are new technologies that have been developed over the past decade, and we were very fortunate to recruit one of the leaders in the field, Ethan Rossi, my first recruit just before Leah Byrne, a few months before. And this is enabling us to detect photoreceptors. And photoreceptors, you can see individual cells in the retina. What you see here are individual cones, and you can even see the rod photoreceptors that are a bit smaller. So we can really image in the living eye using non-invasive technology the status of photoreceptors. And this is very important for the therapies that we want to propose to patients. This is the type of images you can get from an optical coherence tomography. You can extract from the signal the thickness of the photoreceptors, analyze the images of the ganglion cells, all the cells in the retina, and this gives you a very good idea of the health of a retina, not the function, but the structure of a retina. On top of that, it's possible to visualize the photoreceptors. Ethan Rossi has produced this beautiful image where you see individual cone photoreceptors all across the retina. This is the fovea, the very center. And if you move the image, you can even see the underlying cells, the pigment epithelium that are supporting photoreceptors. So you have the ability to navigate through the thickness of a retina and monitor the health of the cells. And this is in the human eye, and it's totally non-invasive and non-aggressive at all. And if you combine all the technique, and this is something that both Ethan here and in Paris, my collaborator Michel Park are doing, you can really image using many technologies, pictures, autofluorescence imaging, where you see the pigment, the optical coherence tomography, where you see the thickness of a retina, and the adaptive optics to visualize photoreceptors. And based on that, as you will see, the decision of potential therapies are going to be made in the near future. So to go back to retinitis pigmentosa, it's a very complex disease. Although the course of the disease is really starting with loss of dark adaptation and peripheral vision, it's the result of many, many gene defects. As of today, and maybe it's already obsolete, we know about 65 different genes. Each of these genes can have hundreds of different mutations. And this is leading to a pattern of disease that is very complex, can be transmitted from different ways of transmission in a family, can be dominant or recessive, or it could be also transmitted on the X chromosome by the mother to the sons. So very different ways. Also, there are many other types of mutations that I don't want to discuss today, but this very diverse group of disease is the result of many, many gene defects. And this makes very complicated the assessment in patients and also the therapeutic decision. For example, recently one therapy was approved for gene therapy, and I think Leah will mention that. It's only adapted to a very small group of patients with early onset disease called Leber's congenital amaurosis type 2. And there are more than 10 types of this disease. And it does really correspond to a handful of patients, but obviously it holds a lot of promises for the future for the others. And currently there are at least 30 clinical trials in gene therapy for many other gene defects. So this is a description of the number of gene defects you can think of. This is not individual genes, these are the functions in the retina. And as you can see, it's very complex. You have many disorders, many functions in the retina that can be affected. And this can give you an idea of why it was so difficult to decipher the cause of the disease and to develop potential therapies. For doing that, we need to do genetic analysis, and we benefit from the expertise of advisors that are genetic counselors. In the department, we have 3 of them. We have Hannah Stanger and Michelle Albeck, and the position that is going to be filled. We're helping every day in assessing the gene defect, and we are talking to the patient and the families, trying to collect the history, the inheritance, all the data we can have, potentially associated disease, and then explain to the patient the results of the test. So this is a very specific function. These people belong to a national society. And in PIT, this is a very important investment that was made by the institution. We have a strong expertise to help. And in my clinic, I'm always working with Michelle or Hannah to really consult the patients on the genetic defect. This is very important because once the gene defect is understood, many therapies can be discussed. Some are targeting rod photoreceptors, some are targeting cone photoreceptors, and they belong to a spectrum of potential therapies. A few years ago, we wrote a review for a top science journal on the potential therapies in the disease. And just to give you an idea, depending on the status of the cells in the retina, the imaging I just showed you the moment ago, we could think about protecting the cells if they are still there and healthy, preventing the loss of the cells over time, or replacing these cells. Protecting the cell, the best is to correct the gene defect, and this is where gene replacement therapy is coming, and this is what Leah is going to focus on, at least in part. When the cells have started to degenerate, the idea is to protect them as much as we can, and this is a very important approach we have been working on for many, many years, both here and in Paris. But once the cells are too advanced to be functional again, then you need to replace them. And these are other approaches like cell therapy, prosthetic, or optogenetics. So we'll try to cover some of that, and we maybe need other webinars to get into more depth. So just we'll talk about gene therapy. And I'm going to hand over to Leah, who is a world expert in that field, to explain to you how we try to cope with this situation.
Thank you very much, Jose. So as Dr. Sahel has already introduced, about 1 out of 3,000 people across the world are affected by inherited retinal diseases. And this group of diseases, it involves mutations in, I think at last count, over 200 different genes. So most of these mutations are actually expressed specifically in photoreceptors, which, as Jose explained, are the light-sensitive cells of the retina, or in RPE cells that provide support to photoreceptors. And as a result of these mutations, there's a dysfunction in photoreceptors and RPE over time, leading to loss of these cells and eventually loss of vision. So it used to be the case that there were no treatments for inherited retinal diseases, but over the past few years, we've seen that gene therapy really holds a lot of promise for treating the entire spectrum of inherited retinal degenerations for the first time. Next slide, please. So in the top row of pictures here, it shows you the process of viral vector-mediated gene therapy. So it works by taking a virus, which naturally has the ability to infect cells and deliver a genetic cargo, and we then replace the viral genome with some kind of a therapeutic gene, such as a replacement copy of a mutated gene. And then we inject the virus into the eye. The virus infects retinal cells and it delivers the therapeutic transgene, leading to expression of some therapeutic protein. So this AAV-mediated gene delivery approach, it really lays a foundation for a variety of different gene therapy approaches, some of which we're going to talk about today. So for example, we could deliver a healthy copy of a mutated gene, or we could use AAV-mediated gene therapy to deliver genome editing tools that directly rewrite a mutation in the genome. Or we could use this to deliver some other type of a therapeutic protein, such as a trophic factor, which would be used to support the survival of retinal cells. Next slide, please. So it's important to mention that the retina is a really promising target for gene therapy. And that is because it's really it's a very accessible part of the nervous system, while at the same time it's isolated from other parts, from other tissues. And so that's really favorable from a safety standpoint. Also, as Dr. Sahel has just shown, the retina is really well suited to functional and structural tests. So we can use these latest imaging technologies like optical coherence tomography and adaptive optics to really evaluate and monitor the progress of gene therapies in real time. And lastly, we understand a lot about the retina. So the architecture of the retina and the function of retinal cells is really well understood. And as we mentioned, we've already identified many of the genes that are underlying retinal degeneration. Click, please. So there are several kinds of viruses that have been successfully used already for gene therapies and for retinal gene therapies, including viruses like lentivirus and adenovirus. But there's one kind of virus that has really emerged as the most widely used viral vector for retinal gene therapies, and that is Adeno-associated virus, or AAV. So AAV, it's a very small, non-pathogenic, non-integrating vector. So it's never been associated with any sort of disease in humans, which is really important. And it has a really proven safety profile through the number of the rapidly increasing number of clinical trials that are ongoing using AAV vectors for retinal gene therapy. So in non-dividing cells, the genome of an AAV vector can be maintained for many years, and that leads to very long-term expression of a gene therapy, potentially for the lifetime of a patient after a single dosage. AAV, it also can infect neurons in the retina, like photoreceptors, really well, which is very important, as some other viruses are not able to infect cells in the retina very well. And another important aspect of an AAV vector is that it's a malleable vector, which means that we can make structural changes to the capsid shell of the virus. And that will influence the way that the virus delivers genes and infects cells in the retina. So I've illustrated in this figure through those yellow portions on the surface of the capsid that we can make structural changes to the surface of the capsid, and that will change the behavior of the virus in the retina. So we can actually engineer new viral vectors that are particularly well suited to be retinal gene therapy vectors. Next slide, please. So as I said, AAV didn't evolve in nature for the purpose of being a gene therapy vector. And so the utility of some of the naturally occurring forms of AAV are limited for some applications in the eye. So some of the current barriers to AAV gene therapy include that they have poor infectivity for some of the cell types of interest in the retina. It's a very small virus, so they have a limited carrying capacity, so they can't carry very large genes. And also they have an inability in some cases to cross the structural barriers that naturally occur in the eye, so that prevents them from infecting retinal cells. So one major focus in the field of gene therapy, and this is something that my lab at the University of Pittsburgh is working on, is to create new viral vectors with improved abilities to infect the retina and to deliver therapeutic genes to the retina. And so one promising strategy that has been employed over the past few years to create new AAV vectors for retinal gene therapy is using this process, which is called directed evolution. So directed evolution allows us to create new viruses with improved ability. And the process works by first creating a really highly diverse pool of different kinds of AAV viruses. So these are slightly mutated AAV viruses. And so each one of these viruses is unique and slightly different from the rest. And then we pool, so that creation, that is called a creation of a viral library. And that's shown in step 1 in this diagram. And then step 2 is to apply some sort of a selective pressure to this pool of AAV viruses. So for example, we could screen the pool, we could apply the selective pressure of being able to infect photoreceptor cells in the retina. And so after this selective pressure is applied to the pool of viruses, it will result in viruses that have the new ability to infect photoreceptors or have some sort of new property that we want them to have. Those viruses will be enriched in the pool of viruses, and unsuccessful viruses will disappear from the pool. So using this kind of a process, we've been able already to engineer new AAV viruses that are much better suited to retinal gene therapy. Next slide, please. So this figure shows some of the data from one such AAV screening experiment. So this is called a scatter plot, and this scatter plot illustrates the performance of a whole pool of AAV viruses. And these viruses have been screened for their ability to infect the retina. So in this figure, every dot represents a different AAV virus. So you can see that there's hundreds of thousands, up to millions of different viruses that have been put through this screening process. And in this particular scatter plot, viruses that performed the best in the screen, or viruses that have the best ability to infect the retina, they have become enriched in the pool, and they make up more of the viruses that have emerged at the end of the screen. So if you could click, Jose. So the most successful viruses in this scatter plot are shown, they're located towards the upper left-hand corner of the plot, and they're shown in red. Next slide, please. So one new AAV variant that has been created using a directed evolution method is called 7m8. So this was a variant that was created through a screen like the one I just described in the context of a mouse retina. And this virus, 7m8, it has an improved ability to bypass structural barriers in the retina and to infect photoreceptors at the back of the eye. So in these images here, you can see expression of a fluorescent protein in a mouse retina following injection of either a naturally occurring wild-type AAV2 or following injection of the newly created 7m8 virus. And what you can see in these images is that there's much more gene expression mediated by the 7m8 variant compared to the parental serotype AAV2 that 7m8 is based on. And this is a body of work performed by a group from UC Berkeley. Next slide, please. So the images that I showed you in the previous slide were from a mouse retina, but of course mice are very different from humans. So they have eyes that are very structurally different from human eyes. Of course, they're much smaller. They have different structural barriers than a human eye. And so for that reason, it was very important as well to show that the virus could deliver genes efficiently to eyes that are very similar to humans prior to using the virus in a clinical setting. So the image here shows expression of a fluorescent protein, a green fluorescent protein, in a primate retina, which is carried into the retina by 7m8. So in this image, photoreceptors are shown at the top of the image, pointed out there by the white arrow. And the blue oval at the bottom of the image is a blood vessel. And then there's red lines that show the inner and the outer boundaries of the retina. And so what you can see here is that 7m8 does in fact have the ability to carry genes to photoreceptors in a primate retina. And therefore, this is a very promising vector for gene therapies in the retina in patients. Next slide, please. And in fact, 7m8 is already currently in use in a few ongoing and upcoming clinical trials, treating diseases like retinitis pigmentosa and adult macular degeneration. And Dr. Sahel is going to be talking more about the GenSight clinical trial in detail later in the presentation. Next slide, please. So as we've discussed, AAV, it's a leading vector for gene therapy, and it's really promising, but it does have some drawbacks. So one of those drawbacks is that AAV is a small virus, and it can only carry genes that are up to 4,700 base pairs long. But some of the genes that are underlying retinal degeneration are larger than this. And delivering just a normal copy of a gene that is larger than this carrying capacity has been challenging. And then there are some diseases for which a traditional sort of gene replacement approach, where a virus just carries in a normal copy of the affected gene, it's not effective for some forms. So for some diseases that are caused by dominant mutations, for example, or mutations that are in parts of the genome that don't directly encode for a protein, those are diseases for which a simple gene replacement strategy may not be appropriate. So for these more challenging forms of retinal degeneration, one rapidly emerging approach is called genome editing. So Jose, if you could click 3 times, please. In genome editing, the portion of the genome that carries the mutation is cut out by a pair of molecular scissors, and then the affected portion of the genome is replaced by the correct genetic sequence. So this is a way to just directly repair the mutation that exists in the genome. And next slide, please. So the tool that's most widely used currently for genome editing is called CRISPR-Cas9, which is shown here on the left side of the screen. So the CRISPR system is made up of 2 components. So the first component is called a guide RNA. It's shown here in yellow. And this guide RNA is basically a homing signal, and it can direct this pair of molecular scissors to a specific location in the genome. And then the second component of this system is a protein called Cas9, which is shown in blue. And that is the portion that can cut through both strands of DNA. So once the DNA that contains the mutation is cut out, then it can be replaced by the correct DNA sequence. And this CRISPR-Cas9 system is most often delivered to tissue by packaging the genes that encode for the system into a virus, which is then injected into the eye. Next slide, please. So another obstacle that is faced by AAV-mediated gene therapy is the fact that, as Jose said previously, there are a large number of mutations underlying retinal disease, and there are many forms of inherited retinal degeneration, many types of mutations leading to retinal degeneration. So it's actually a challenge to create gene therapies and to conduct clinical trials for the many forms of inherited retinal degeneration that exist. But one very promising approach to treating many forms of inherited retinal degeneration is an approach called trophic factor gene therapy. So the basis for this trophic factor gene therapy lies in kind of understanding the mechanisms by which cells are lost in retinal disease. For example, one large set of inherited retinal degenerations are rod cone dystrophies. Retinitis pigmentosa is an example of a rod cone dystrophy. So in rod cone dystrophies, there are mutations that are specifically expressed in rod genes. And so during the course of this degeneration, there's a first phase of the disease where rods are lost, and the patient experiences a loss of night vision and peripheral vision. However, there's also a second phase of the disease in which cones, which are the cells that we use for high acuity central and color vision, are lost. And so this was somewhat of a conundrum for a period of time. And the question was really, why are cones dying from a mutation that is specifically expressed in rods? And it turns out that one of the main reasons that cones die after the loss of rods is that rods supply a survival factor that cones require. So this factor, which was discovered by Thierry Leveillard and by Dr. Sahel, is called the rod-derived cone viability factor, or RdCVF. So when rods die, the source of RdCVF is missing, and cones begin to die. Next slide, please. So understanding this biology underlying the loss of cones and rod cone dystrophies really lays the groundwork for a very potentially powerful approach to treating the wide variety of forms of rod cone dystrophy that exist. And we can do this in sort of a mutation-independent approach. So supporting the survival of cones can have could have dramatic benefits for patients. And it's been shown that 50% of cone loss can be compatible with 20/20 visual acuity. And it's been estimated that a cone preserving type of treatment or approach could prevent 1.5 million people across the world from becoming blind. And that's because in modern society, we mostly use cones for our daily activities. Next slide, please. We've tested AAV-mediated delivery of RdCVF in animal models of blindness, and we've seen that RdCVF treatments can preserve cone function in mouse models of rod cone dystrophy. So here on the left, you see images from a mouse model of retinitis pigmentosa, and cones in these images are labeled in red and in blue. And so this image really illustrates that in mice that were treated with RdCVF, cones can survive longer than in animals treated with a control injection. And then in the right panel, on the right side of the screen, these are electroretinogram recordings, which are recordings of cone function. And so they record the function of the retina in response to a flash of light. And the first set of tracings on this panel are from mice injected with AAV RdCVF. And you can see that they have a larger retinal response than animals that were treated with controls. And there's tracing from a wild type mouse is on the very far right of that panel. Next slide, please. So just to summarize this portion of the talk before I hand it back over to Jose, I just wanted to summarize. So we've discussed that gene therapy involves delivery of a therapeutic gene to retinal cells. And we've talked about how AAV vectors are safe and effective gene delivery tools and a leading vector for gene therapies. And a gene delivery platform can be leveraged for many different types of gene therapy approaches. And that those include gene replacement approaches or genome editing approaches, directly rewriting mutations in the genome. It could also be used for delivery of trophic factors to support survival of cells in the retina, and as well as many other approaches that we didn't really have time to touch on today, including things like optogenetics, creating artificial photoreceptors in the eye, that Jose will be talking about in more detail later as well. So together, we're really aiming to develop all of these different approaches in order to create a toolbox, a gene therapy toolbox that has therapies that could be appropriate for the entire spectrum of retinal diseases, and therapies that could be applied to any phase of a disease. And so with that, I'm going to hand it back over to Dr. Sahel.
Joe's, oh, there you go.
Yeah. I'm going to continue to discuss the other therapies that are made available. So I'll start with the potential to restore some vision in patients that lost most of the cells. What you heard from Leah is a group of approaches to correct the gene defect at the initial stages, to protect the cells when the process of degeneration is ongoing. But once you get to a stage where photoreceptors have disappeared or are impossible to rescue, what can we do? And so among the approaches is a retinal prosthesis. I won't spend a lot of time on that because in the previous webinar on age-related macular degeneration, we showed the amazing progress that has been made in the field. The concept is that in the remaining cells in the retina, photoreceptors having disappeared, you still have the rest of the cells in the retina, all the neurons that are connecting to the brain and providing the information to the brain. But the light is no longer captured by the photoreceptors because they have disappeared or degenerated. So two approaches have been undertaken in the past decade. One is to stimulate the ganglion cells themselves, the same cells that connect to the brain. And this technology was the Argus technology that has shown some success in Paris. We have done many patients with that, and there are many patients that were treated in the US. But this technology was discontinued for commercial purposes, but also because the resolution was not very good. We worked on another approach, which is to replace the photoreceptors with a panel of electrodes that are sensitive to light, replacing photoreceptors. So this is what we have developed in Paris in partnership with Daniel Palanker at Stanford, who got this initial idea, and Vision Vision in Paris. And this is a chip that is formed of diodes that are sensitive to light. It's like a solar panel that is responsive to light and is inducing electrical current. It's placed under the retina during surgery and replaces photoreceptors. So we move that from the preclinical to the clinical. This is an image of a patient that was operated in Paris, and actually 5 patients have been operated in Paris, and the only site in the US that has been able to do that is now Pittsburgh, with 2 patients that have been treated, and this is ongoing. It was a bit delayed by the COVID crisis, but this is ongoing. So Dr. Martel reported on that, and this is currently on age-related macular degeneration, but it has the potential to apply also to retinitis pigmentosa. And the level of vision that is afforded to that is the ability to read big letters or words, but obviously not normal vision. So this is very promising. I won't spend too much time because we already discussed that, and there is more to come. And the trial is ongoing in Pittsburgh, which is currently the only site doing that. The other approach is to transform the remaining cells in the retina into photoreceptors. It's a technology called optogenetics, very complex technology. We have been working on that for the past 15 years, and the concept is the following. In the mammalians, including humans, mice, primates, we have a very complex system in photoreceptors, hundreds of proteins that are responding to light and triggering electrical signal. If any of them is mutated, then we have a disease like retinitis pigmentosa. In several species, very simple species actually, like parasites and a very simple organism, there is another system, which is one protein that can capture the light and trigger immediately an electrical signal. This is very simple, this is very robust. Obviously, this is less light sensitive because the system we have in mammalians is amplifying the signal by 1 million. We don't have that, but it is a very simple system. So the idea is to express these proteins in remaining cells in the retina. And this is what we have done using optogenetics. We can either put back some protein sensitive to light in the cones once they have lost most of the seeing part of the cones, or you can trigger the remaining cells in the retina, like the ganglion cells or the bipolar cells. So this is what we developed here. There was another group in Detroit that worked on that too. And this is the technology we have been developing by GenSight technology. We need to couple the system, we need to express in the retina the protein that is sensitive to light, but because you need a significant level of light and you need an amplification of a signal, we develop goggles, and these goggles are like a projector sending the light into the retina and stimulating the remaining photoreceptors. So this is a technique that was developed conjointly at the Vision Institute, and this technology is now in the clinic. One of the scientists who is at Pittsburgh, Riyad Ben Osman, contributed to the design of these goggles, and it's a combination of a system of stimulation and gene therapy. The trial is in Pittsburgh, so we are using the vector that Leah was showing a moment ago, is being used currently in Pittsburgh, and also active in London and in Paris. We predicted the type of visual acuity you can get. This is studies that were done in the retina in a petri dish, and these are projecting letters to the retina and calculating the signal, and we predicted a visual acuity which would be above the threshold of 20/200, so which is the threshold of what we call legal blindness, being able to read letters and words. Another approach which is promising, and you read too much of that in the news, and a lot of charlatans are proposing that. Just as a reminder, anytime you are asked to pay for a therapy that is still in a clinical trial, just go away, because it means that this is not an approved therapy. This is just something that people want to make money out of it, because all the trials should be free. Nobody should be asked to pay anything for a clinical trial. And stem cells are very complex. You need to replace the cells in the retina. We have been working on that also. And I just show you an example of what we have been able to do. from cells that are collected, for example, from a piece of skin where you are able to generate in vitro all the cell types in the retina, the underlying cells, but actually a full retina in a petri dish. It's possible to identify all the cell types in the retina and to transplant them. And just to give you an example of a technology, this is in a mutant animal. This is the normal optical coherence tomography. This is the animal that is not treated, the cells have degenerated. And this is the treated animal with transplantation of cells that we developed in Paris and that we would like to transfer here. You see that there are many more cells rescued in the treated retina versus the untreated with this technology. So this is now in clinical trial in Paris. And these are the cells that have been transplanted in the patient. You see this layer of cells and they are alive. It's already a significant time. So this is just to give you an idea of the number of therapies that are in development, and I could spend a lot of time on each of them. Each of them is very promising. But what you have to remember that there is no cure for everyone. Each cure has to be personalized. Many of the genes are still not targeted by gene therapy, and many patients are in very advanced stages. But using the imaging technologies that I showed a moment ago, we can determine the status of a retina and try to define what could be the best possible therapy for the patient. So very complex, but so much progress made in the past few years. But then the next question is this helping people? Is this changing their lives? People that are so strongly visually impaired, are they benefiting from that? So we decided to explore that, and this is an activity that I started in Paris that is now happening in Pittsburgh. We developed a system to analyze the vision, not only in the clinic as classically, but also at home. So this is a home that is mimicking what happens at home, but also an artificial street. And I'll show you what we have been doing. This is called the street lab. We are asking patients to perform the task of daily living. They tell us what is difficult for them in daily life. We put captors that are working in infrared to determine the movement of a patient, and we are trying to detect the impact of visual impairment in daily life. To give you an example of what it means, this is a patient that has a very narrow visual field. The patient is moving in this environment, and we are following the direction of the gaze. We are following the gait of the patient and the trajectory of the patient and trying to determine what happens when you have such a narrow visual field that you cannot see everything, just a small part of the environment, how you avoid obstacles, how fast you can perform, and how you are able to perform. This is done with mobility, but it can be done also with grasping objects, recognizing objects, for example. And just to give you an idea of how it is useful, I'm going to show you what we have done with a patient that have been treated with the first approved gene therapy. We did the largest number of patients in the world in Paris with that. And this is a kid that was blind from retinitis pigmentosa, and she has to perform in very low levels of light, and she has to navigate through a maze, this type of street that we developed and I showed you. And as you will see, it's a bit long because the kid is really not able to follow the lines. And these are the dots, for example, she has to follow. And it's taking her very significant time. She has very limited vision and she's 9 years old. She has been in that situation. So she just cannot see. And she has to find out what would be the crossing path through a road. And it takes her a very significant amount of time. So we are monitoring that very carefully. And as you will see, it takes her a lot of time, a lot of inaccuracies, and a lot of obstacles that she's bumping into. This is after the gene therapy. This is what's happening. The same kid a few weeks later, and she's now moving almost normally in that space. She's doing a lot of activities. She's going back to normal school. And I could I have a lot of images of many patients that have been treated this way. And this is extremely promising for all these patients. So it tells you that you have to do these metrics to measure. So just to summarize what we try to show today, we are talking about a disease that is destroying photoreceptors over time. When photoreceptors are still there, gene therapy or gene editing, as described by Leah, could potentially restore normal vision. But many patients come too late, and many patients cannot be treated before the cells have degenerated. This is where neuroprotection with trophic factors can protect the remaining vision, the 20/20 central vision that people want to keep. When it is further down the road, we can still hope from prosthesis, cell therapy, optogenetics to restore some form vision, not perfect, but ability to recognize faces, hopefully, emotions, and reading. From all of that is driven by understanding better the status of a retina and the potential for therapies. So it's really very promising, but very early. I just want to thank the many people who worked on that in Paris, and here the funding agencies like Foundation Fighting Blindness, the NIH, and others, and obviously many people I cannot even mention. I want to tell you that this is going to be at the core of what we want to develop in the new building with the support from the INDF Foundation and all of you. This is part of the integration between science in the upper floors and clinical activities in the middle floor, and really delivering care to patients. We are just at the beginning, at the steps. We have just done a lot of work, which is just bringing us to what Winston Churchill called the end of the beginning. We are still in the beginning, working on it and trying to propose to you the best possible therapies for the future. So thanks a lot for your attention, and hopefully there is much more to tell in the next few months and years. Thank you.
Oh my goodness. Well, thank you so much, Dr. Sahel and Leah Byrne. This is, it's a lot of information, and it's all incredibly, incredibly impressive. I'd like to open up. We have a few questions already, but please put your questions into the Q&A and we'll start reading those. I think that, I'll start in a sense with some things which I know people might have a hard time understanding. I mean, it's mind-boggling that viruses can be used this way to be able to go in and edit a gene. And Leah, you did talk a little bit about and you showed a very good example of how that's done with the gene editing tools, but it still is mind-boggling that you can do that and essentially apply like a pair of scissors to edit a gene. So we'll have to take your word for it as far as how that actually works. But can you, is this something that you feel for, for all these different types of retinal generations in the future, that there's possibility to just significantly improve from, and what's the potential for intervening with gene therapies at a young age for someone who we know tests positive for genes that would ultimately lead to blindness?
So certainly there is a great deal of potential for all of these therapies, for all of the approaches that we've mentioned here today. I would say that, as Jose mentioned, I do think that we're at we're not at the beginning, but there's certainly a lot of work to be done. But all of these strategies have a lot of potential, including genome editing. So some of the major obstacles that we're facing with genome editing are definitely efficient delivery to cells, which is something that my lab is working on. And obviously we need to mitigate the safety concerns as much as possible as well. So for genome editing, one thing that we're working on is making sure that that editing is extremely specific, so that you're only editing exactly the part of the genome that you want to edit. So there's definitely work to be done, but I do think that the potential of all of these therapies is tremendous.
I can imagine, and I think I asked the question before one of our attendees did, but someone here says, "Bonjour, Dr. Sahel. Do you have an estimate on when RP transplant could be available for patients? I have a degenerative disease and love to get treatment before my cells die." I think that.
So before I get to that question, I have to say that we have already several studies using gene therapy in the department at the clinical stage. The optogenetics study is ongoing. Corrective gene therapy, we have ongoing trials, and we are planning to get the approved therapy also in Pittsburgh. We are working on that very heavily currently. So all of this is very promising. In terms of cell therapy, we are very careful with that because we don't want to overpromise. So the trial just started in Paris last year, and we would like to bring the technology. Other sites in the US are working on some cell therapies. We believe that it needs to be a very structured approach with a very organized tissue, because you don't want these cells to proliferate in the eye. So it's a matter of probably a few years to get that in the clinic. But when we say a few years, very often it's a few years for for a clinical trial. For an approved therapy, it takes longer than that, and we want to do everything very safely. We don't want to take any risk. The last thing we want to do is to compromise the chances for a patient to benefit from a better therapy down the road. So we do that step by step. But this is ongoing, and this is actually, as you know, I'm very impatient in nature, so we try to make this happen as soon as possible, but as carefully as possible also.
Well, I think this partially answers the question, but maybe this is a little more for Dr. Byrne. Well, how long until the virus therapy will be in use at UPMC Eye Center on a regular basis? Essentially, when we'll get through clinical trial.
Right. So as Jose mentioned, there are clinical trials that are. So there's one gene therapy that is already clinically approved. That's this drug called Luxturna, which is a gene therapy for RPE65, Leber's congenital amaurosis type 2. We already have many other clinical trials in motion for AAV-mediated gene therapy, and many other clinical trials in the planning stages or almost ready to begin. And I think that the number of clinical trials is just rapidly increasing. And there's also a lot of industry participation in gene therapy right now. So this is a rapidly advancing field. And so it's a little bit difficult to predict exactly when things will be clinically available, but certainly within the next few years, I think we're going to see a lot of different forms of gene therapy available to patients.
Are there limitations to the AAV therapies? Are there certain groups that this may not likely work for?
So there are certainly limitations that we're working hard to overcome. And I think I briefly touched base on a couple of them. So some of the major obstacles that are preventing gene therapy from being efficient are that there is currently an immune response to the AAV vector. So as I said, AAV is a non-pathogenic virus, and so you would never know if you'd already been infected by the virus. And if you have been infected by the virus previously, then you have likely generated antibodies against the virus, which would potentially make the gene therapy less effective. And so one major area of research in the field of gene therapy is overcoming that immune response to the viral vector. And so if you have an immune response to the virus, then it might be that we need to come up with a different sort of a strategy that would work for you. So already there are some approaches to overcome that. You can administer the virus to different compartments in the eye. One of those compartments is called the subretinal space. And we know that injecting virus into the subretinal space largely protects the virus from the immune response. So there's a lot of different approaches to overcoming that particular obstacle. I briefly mentioned that the carrying capacity of the virus is limited too. And so right now, the field is coming up with a lot of different creative approaches to overcoming that limited carrying capacity. One promising approach is, so if you have a gene that's too big to fit into the tiny AAV virus, groups have been trying to divide up that gene into two different pieces that are delivered by two different viruses. So that's one example. As we talked about as well, so we're developing other strategies that would be useful for different phases of the disease progression. So if a patient is at a phase of the disease where photoreceptors are already missing, you can't go in and protect those photoreceptors from being lost. You would need a different type of approach, something more like an optogenetic approach, where you create photoreceptors out of different cells in the eye that are still remaining. So I would say that there are currently obstacles, but that the field is already thinking very creatively and working very hard on strategies to overcome each of the obstacles that exist. And so it's going to take some more hard work and creativity on the part of researchers in the field, but there's a lot of really intense effort ongoing right now.
It is interesting. You can't help but think, as we're talking about viruses, that we're in a pandemic, and it kind of sounds frightening to talk about viruses that you actually purposely stick in your eye. But
it's important to remember that the world is basically awash in virus, and most of the viruses do not create a disease like coronavirus. Yeah.
Yeah, and it's interesting. I know we talk about this, and how you refer to it is really like essentially like a Trojan horse that goes in and delivers what you want to deliver, and it's a fascinating tool. I'll just touch a quick question on optogenetics, as I think that this is something that's just so fascinating. I understand how this works, and essentially you're providing a photoreceptive cell that's otherwise been lost, and you're providing a cell that ultimately is available in nature. But how on earth, and I guess this is directly to Dr. Sahel, did you come up with this idea that this would work and be something that could be done? It's remarkable.
So the idea came from a group of scientists in Germany in the early 2000s, actually, when they identified initially in a parasite and in a microorganism these proteins. And they got the idea that you can manipulate neurons. And early in 2000, it was qualified one of the discoveries of the year to really be able to manipulate neurons and to activate neurons. And the idea to activate the cells and to make them sensitive to light came in parallel to a few groups. One group in Detroit, our group in Paris at the time, and Boton Roska in Basel. We started to collaborate on that. So it's the idea that you are losing the ability to respond to light, and there is this protein that is responsive to light. So it's a pretty simple idea. It's more complex to find how to activate it, to modulate the signal. So, but in principle, this idea is once you get the protein, I think it's quite an obvious one. Just using nature to cure nature. It's a natural solution.
That's amazing. Well, thank you. We are at the end of the hour, and this is a really wonderful program. We're happy to share it with everybody in the audience today. This will be recorded and available on our website in a few days. If there are more questions, please feel free to send those to the email that was provided for you in your invitation to Mr. Craig Smith, and we'll certainly get those to the experts to answer. Once again, thank you for joining us for the Eye and Ear Foundation biweekly series, Sight and Sound Bites. Have a great day.
You too.